Hi everyone, welcome back to my YouTube channel Biology Medici. In my previous video that I released last week, I talked about and introduced you about the protein sorting through endoplasmic reticulum. Today we will talk about the import of proteins into the mitochondrial matrix. Now, mitochondria are considered to be the powerhouse of cells. There are numerous proteins which are, although mitochondria have their own ribosomes, but there are numerous proteins which are synthesized in the cytosol and they need to enter the mitochondria. Therefore, there must be a way which exists using which the proteins can enter through mitochondria. Now, as you have come to know, the basic principle of protein sorting is the presence of a signal peptide which is present either in the C-terminal end or in most of the cases that is present on the N-terminal end of the polypeptide concerned. As you remember, we talked in the last video, for example, the signal sequence of those polypeptides or proteins which will enter through the endoplasmic reticulum, they are present on the N-terminal of the polypeptide chain. Now, this sequence must be removed once the polypeptide enters the endoplasmic reticulum. And you know that there are certain characteristic features which are present on this signal peptide, and then comes the mitochondria, which we are going to discuss today. Now, in case of mitochondria, the signal peptide is present on the N-terminal end, and this signal sequence must be removed as it happens in case of the endoplasmic reticulum. Now, talking about the features of the signal sequence which is present in the mitochondrial peptide, this sequence consists of 20 to 50 amino acid residues, and at one of the ends there are arginine and lysine which are present, and on the other side there are hydrophobic residues present on the signal peptide. Therefore, compared to the endoplasmic reticulum signal sequence, the mitochondrial signal sequences are a little larger. We do not know the reason yet. Now the question comes, what is the mechanism of importing this protein from the cytosol into the mitochondria? You see, the protein must be unfolded in order to enter through the mitochondria. So either these proteins remain unfolded or they are partially folded. So they can never be folded. So folded proteins cannot enter the mitochondria. Now the question is how they are rendered unfolded. So there are certain chaperones proteins which are present in the cytoplasm of a cell, and these chaperone proteins, for example HSP70, they render this protein unfolded at the expenditure of ATP. Now these unfolded or partially folded proteins are captured by certain import receptor. In this case it is TOM2022. So TOM stands for translocon of outer membrane. Now upon capturing this particular polypeptide, now this TOM2022 protein come closer to a general import protein, and then this polypeptide is transferred to this general import protein. Now you see at this particular position the inner membrane and the outer membrane, they tend to touch each other. So that means there are certain rare contact sites at mitochondria where the inner membrane tends to touch the outer membrane. So this polypeptide can easily be now channeled through the TIM44, right? And this TIM stands for translocon of inner membrane. So as a consequence, this polypeptide now get entry into this particular translocon protein present on the inner membrane of the mitochondria. This polypeptide is now pushed through the translocon protein at the expense of ATP. So upon entering into the mitochondrial matrix, now the signal peptide is cleaved, and then these signal peptides can be folded to assume its native conformation. So that's all about the import of mitochondrial protein. If you are watching my videos and think that these are useful to you, then please like the video, share it, comment in the relevant section and subscribe my channel. And please do not forget to press the bell icon in order to get the update of newly released video. Thank you for watching.